The AQA essay is worth almost 10% of your final A-level grade, so preparing for it thoroughly is essential for success, and that's exactly what I'm going to be helping you to do in today's video. Hi everyone, and welcome to Miss Estrick Biology. And if you are new here, then I've been teaching for over 14 years, and in this video I'm going to be sharing with you 3 obscure essay titles that are similar to some that have come up, but not in a long time. So I think they'd be really good ones to plan, to prepare for in case they came up or something similar to them came up. And even if they don't, it's still great practice of planning the essay and also revision of a range of topics. Now, you might have seen my first video where I went through 3 essay titles as well. And I'm going to go through the same disclaimer that I went through last time. Now, I don't do predicted essays. Now, I know last year, the video that I did on this, I had 3 essay titles, 2 of the 3 are the ones that came up. And I do pick the essays that I plan with you based on lots of analysis that I've done. So I have analysed every single essay title that's ever come up on the new spec, old spec, specimen papers, look for patterns and do other things to try and get an idea of what might come up each year. But I don't have any insider knowledge, and it doesn't mean that what I say is what's going to happen. It just so happens to be that in the last couple of years, a couple of the essays or one of the essays that I did talk about did come up. But that doesn't mean that the essays that we're going through in today's video are going to come up. So make sure you still revise all options and not just put all your hopes on the fact that it could be one of these. And as I said, I've picked 3 titles that are slightly more obscure. One of them in particular I think would be too narrow a title to come up, but maybe something similar would. So that's what I'm going to be going through with you today, the 3 essay titles to have a go at. I do also want to say a massive thank you for your patience, because you might have seen on Instagram that I'd been ill for a couple of weeks, and that meant I had to delay my free essay masterclasses. Now, those have happened now. Well, I say they have happened. They've happened for you. They've not happened for me yet. They're happening in a couple of days, but by the time this video's come out, they would have happened, which means that my biology essay bootcamp is now live. And this is an online course which is made up of 6 lessons, everything you could possibly need to help you to get full marks on the essay. So if you do still need more help, then check out this course. It's designed so that you can complete it before your exams, but it gives you everything you would need. So it's got lessons on the mark scheme, how to write the essay, topics you could apply to any essay, beyond the specification information. I help you plan essay titles. I also give you a model essay as well. So if you want to find out more about that, then click the link in the description. But for now, let's get into those 3 essay titles. So the 3 essay titles are: number 1, the importance of carbon-containing substances. Now, this exact title has never come up, but there was a title that used to come up quite a lot on the old spec, which was the importance of carbon dioxide. And they've not used that since, because you now no longer have to learn about the carbon cycle. However, I did think maybe they could tweak that title and have it the importance of carbon-containing substances, because there's loads of biological molecules that you learned about. So that's one title that we're going to go through. The second title is the importance of hydrogen bonds in organisms. Now, this is the one I think might be slightly too specific to come up, but I picked it because it did actually come up 14 years ago on the old spec, and it's quite similar to the essay title that came up in 2019, which in my head I was like, that's not that long ago, but it was actually 5 years ago now, so it's quite a long time since the title, the importance of bonds and bonding, came up. Now, that's obviously a lot more broad because you could talk about any bond. For this one, I narrowed it down just to hydrogen bonds, which in reality is probably too specific, but I still think it's really good revision and a worthwhile one for us to plan together. The final one is the importance of receptors in organisms. Now, this has only ever come up on the specimen paper for the new specification, and I quite liked this title because receptors could be either cells that detect a stimulus, so it could link to what you learn about in the nervous system and homeostasis, but receptors are also binding sites, like the unique 3D shapes. So there's a whole range of different things you could talk about for receptors. So I really liked that as a potential of an essay that has such a broad range. So watch me plan these, and I'm going to talk you through it as I plan each one on the whiteboard. So here's the first essay title, the importance of carbon-containing substances. Now, I'm going to plan 5 paragraphs. These aren't the only 5 that you could talk about, but the key thing is making sure that you are always picking titles or topics that are relevant. You can link it back to the fact it contains carbon and making sure it's not from literally the same subsection of the specification. So I'm starting with glycogen, but because I've got glycogen, that is from the polysaccharide section. So I couldn't also talk about starch and cellulose. So for glycogen, we need to say our AO1 first of all. And for my AO1, I'd be talking about the structure of glycogen and trying to demonstrate as much A-level knowledge as possible. So I'd link to the fact that I know the monomer is alpha glucose. I'd be talking about the fact that there's 1 to 4 and 1 to 6 glycosidic bonds, but it's the 1 to 6 glycosidic bonds that result in that branch structure, and it's a highly branched molecule. I would then talk about the fact that it's so highly branched, that means that there's a large surface area for enzymes to attach to if glycogen needs to be hydrolyzed back into glucose. I'd probably also point out that you have a large store of glycogen in the liver and muscle cells. Then if I start to link that to why is glycogen so important, you could talk about the fact that you can fit a lot of glucose in a small space. It's insoluble, so it's not going to affect the water potential of the cell. And the key one, as we said, is it can be rapidly hydrolyzed back into glucose, and that glucose can then be used for respiration so that lots of ATP can be made and energy from that ATP can be used for metabolism. So that would be my concept of my AO1 and my AO2 for glycogen. The next one then is proteins. So I'd start by describing protein structure, in particular pointing out where the carbon is. Now you can actually do diagrams in the essay, so I'd probably draw the general structure of an amino acid in my essay, so I could then point out it contains a central carbon, and also there's a carbon in the carboxyl group. And then I have ticked off that it's a carbon containing substance. I would then talk about the fact that peptide bonds form between the carboxyl group and the amine group of two different amino acids, and through multiple condensation reactions, you then end up with your amino acid sequence or your primary structure. Then I talk about my secondary structure, how it's the folding and held in place with hydrogen bonds to get either that beta-pleated sheet, or if it's coiled, you get that alpha helix. Then further folding and the introduction of ionic bonds as well now, disulfide bonds and more hydrogen bonds results in that unique 3D shape, the tertiary structure. You could also mention that you could have a quaternary structure, which is when you have more than one polypeptide chain. Now I'd then link to the importance of that unique 3D shape in regards to enzymes. So I'd talk about the fact that you get that unique 3D shape which creates an active site which is almost complementary in shape to the substrate, and link it to the induced fit model to then explain how enzymes are lowering the activation energy required for a reaction to occur and how this speeds up reactions. And then you could link it to just one example, but don't go through too much detail. You could just say, for example, helicase and DNA replication. You don't need to go into extensive detail because my focus would be more explaining the induced fit model. So that would be my proteins paragraph. I've described the structure of proteins, linking it to the carbon containing substance because you have to keep linking it back to the theme of the question. And then I've said why that protein is so important, applying it to the example of proteins. Again, linking it back to the title, the importance of. My next paragraph then I would do about is DNA. Again, I'd potentially draw a DNA nucleotide so that I can show that we have carbon in that pentose sugar, deoxyribose. And then I would talk about the condensation reactions between that nucleotide to form a polynucleotide and how you then have two complementary chains held together by hydrogen bonds to make that DNA double helix. But because I'm going to link my importance to protein synthesis, I'm probably more going to focus on the fact that within one polynucleotide chain, you have this unique sequence of DNA bases. And then when I get to the importance of, I can say the importance of this sequence of DNA bases in a gene is that it will code for a unique sequence of amino acids to create a unique protein, which has a particular function. So that's how it then lead into my AO2. And I wouldn't go through lots of detail on every single part of protein synthesis because that's when it can start to become a bit irrelevant. So the main thing would be transcription. transcription, because transcription is the stage where the DNA is literally used to create a copy in the form of mRNA. So I'd talk through A-level standard details on transcription, and I've got a whole video on that if you want to see the exact details to include. I'd then briefly say that the pre-mRNA would have introns removed. It then leave via the nuclear pore to the cytoplasm, attached to a ribosome, and then through translation you'd get a polypeptide chain, or that unique sequence of amino acids. And I'd say this is important because it means the DNA base sequence has coded for a unique sequence of amino acids, which will ultimately determine where bonds form, the final 3D shape, and therefore the function of a protein. And that's how I then wrap it all up, making sure I've linked it constantly to the carbon containing substance, which is the DNA in this case, and why it's important, because it codes for that exact amino acid sequence. Next one I'm going to go for is ATP. Now for ATP, yet again, I'd probably draw the molecule. So showing that you have 3 inorganic phosphates attached to ribose, and then attached to that, you'd have your adenine. And ribose is a sugar, it's a pentose sugar which contains carbon. So that's how I'm linking it to, it's a carbon containing substance. I would then give some details on the fact that if you hydrolyze ATP using ATP hydrolase, that's going to break the bonds between the phosphates, and that releases a small amount of energy. I'd say it's small manageable amounts of energy, which can then be used for metabolic processes. And this is when you could then lead into your importance of ATP and give a particular example. So you could maybe talk about its importance in the terms of binding to myosin head in the sliding filament theory to enable the myosin head to detach, recock, and then you can get it attaching again and the muscle continues to contract. Or you could talk about its role in the Calvin cycle, because it provides the energy for the reduction of 2 molecules of GP into 2 molecules of triose phosphate, and also it provides the energy to regenerate RUBP. But there's loads of different examples, but that's what I'd have as my importance of for ATP. I would talk through a particular example that you learn about where ATP is providing energy to demonstrate its importance. The last one that I would go through is lipids, and I'm going to go for phospholipids. So you could describe the structure of your phospholipid, the fact that you have 2 fatty acid chains attached to 1 glycerol molecule, and then you have an inorganic phosphate attached to that glycerol as well. I would then talk about the different regions of that phospholipid and the different properties. So the fact that the tails, which is where you have those fatty acid chains, are hydrophobic, meaning that it won't interact with water, it'll repel water, but it can interact with fats or lipids. Whereas the head structure, where you have the glycerol and the inorganic phosphate, that is hydrophilic because of the charge on that phosphate group. And that means if it's hydrophilic, it can interact with water. And it's this hydrophilic and hydrophobic region on a phospholipid that enables the formation of the phospholipid bilayer. This is then where I'd link it into the importance of, although actually I haven't actually mentioned, I would also make it very clear that glycerol contains carbon. It's a 3-carbon compound, and the fatty acid chains is a hydrocarbon chain. So that's where the carbon is within that molecule. So you always have to link it back to the theme of the question. The importance of, which is also linking it to the theme of the question, but that's what we call our AO2, so applying your knowledge. That one, I would then be talking about how it creates that partially permeable membrane, and only lipid-soluble molecules can dissolve through the phospholipid bilayer. So that would be my plan for the first essay title. The next essay title is the importance of hydrogen bonds in living organisms, and I'd start with hydrogen bonds in DNA. I would link this to the fact that you have hydrogen bonds forming between the 2 polymer chains in the double helix, and you have 2 hydrogen bonds forming between adenine and thymine, and 3 hydrogen bonds forming between guanine and cytosine, and how these hydrogen bonds hold the chains together and collectively provide strength, but individually those bonds are quite weak and they are easily broken by the enzyme helicase. Then that would link into my AO2 importance. That is important because you want to be able to separate those 2 DNA strands so that they can both act as a template in DNA replication. Give a little bit of details on DNA replication and say that this is important because it means you can get identical copies of DNA in new cells. So in processes such as clonal expansion in the humoral response in immunity, that means you get lots of copies of identical B cells, which will all be able to produce the same antibody. So that would be my linking it to the hydrogen bonds and then the importance of. Next then is cellulose. And with cellulose, I'd be starting by describing the structure. So beta glucose is the monomer, 1 to 4 glycosidic bonds. You get long straight chains of beta glucose, which align in parallel, and those are held together by many hydrogen bonds. I would then point out again that the individual bond is weak, but because there are so many of those hydrogen bonds, collectively it provides strength, and that's what a fibril is. It's lots of long chains of beta glucose held together by fibrils. This provides structural strength to the molecule, which is found in the cellulose cell walls of plants, and this would then link to my importance of. This is important because it provides that structural strength to a cell wall, so that when water moves into a plant cell by osmosis, when the cytoplasm of the plant cell would have a more negative water potential, it would mean that water would move in and the cell would become turgid, but the cell wall won't burst. So that would be how I'd link it to the AO2. I'd probably have a big focus on the idea of water potential to make it A-level standard as well. Next then is water, and I would describe the polar structure of water. Potentially I would draw 2 water molecules and show the delta positive on the hydrogen, delta negative on the oxygen, and that hydrogen bond can form between the oxygen and the hydrogen of 2 different water molecules. And that would then link to the fact that it gives water this cohesion, meaning different water molecules stick together because of all those hydrogen bonds that form between them. And this enables water to move as a continuous column up through the xylem during transpiration. And that links to your cohesion tension theory. So you could talk about all of that for your AO1. My AO2 of why that is important, I would then link to the fact that it enables transpiration and transport of water from the roots, where it's absorbed, to all of the cells in a plant. This is important in particular for the leaves and the palisade cells, because that's the site of photosynthesis, and photosynthesis involves photolysis or photolysis in the light dependent reactions, which is the splitting of water. And if we didn't have that stage occurring, then ultimately you wouldn't get those organic molecules being made, which the plant needs to make glucose for respiration. So we've gone through the hydrogen bonding and why it's important. Proteins is another topic that I would talk about. And for this one, similarly to what we said for the carbon containing substances, I would describe the structure, but this time have more of a focus on the secondary structure and the tertiary structure rather than creating that polymer chain, because it's the secondary structure, that alpha helix and beta pleated sheet, that's held in place by the hydrogen bonds. And you then have even more hydrogen bonds in the tertiary structure, along with the ionic and disulfide bonds, to give that unique 3D shape. When you then get to the importance of, that is going to be exactly the same as I said in the previous title. So I'm going to do enzymes, induced fit model, lowers the activation energy, and then maybe say an example. So for example, I don't know, you could pick a digestive enzyme maybe this time. So you can start to see that the more essays you plan, you can recycle and reuse paragraphs that you've used previously. And that's why I think doing lots of plans is such a helpful revision tool, because then you can pick and choose something that you've done previously and maybe slightly tweak it. So when you get to your actual exam, you should be able to write a really good essay. The last one I've got is tRNA. Now tRNA, I would talk about the fact it is a single strand molecule, but it folds into that clover leaf shape held in place by hydrogen bonds. And that clover leaf shape enables you to have that amino acid binding site at the top and the anticodon, the 3 bases exposed at the bottom. That means that tRNA molecules can bring specific amino acids determined by their anticodon, which is then going to match up to complementary codons on mRNA. When I then link that to why is it important, you can talk about the fact that it ensures that tRNA molecules bring specific amino acids to the codon, so you get that unique amino acid sequence, which then results in the correct or unique shape 3D structure protein being made so it can perform a particular function. And then we get to the final one, the importance of receptors. Now for this one, I would start with Psinium corpuscle. So I'm going for a receptor, which is one that detects a stimulus. So I describe the Psinium corpuscle, talk about how it works. So thinking about all of those layers of lamellae wrapped around a sensory neuron, and how when pressure is applied, it stretches those membranes, and therefore it pulls apart those stretch mediated sodium ion channels, opening them up. So sodium ions diffuse in, that then results in depolarization, and if you have enough sodium ions moving in that you meet that minus 55 millivolt threshold, then an action potential can be generated. Then you can link to the importance of that. This is important because it enables you to respond to changes in your environment, for example, pressure, which might mean that something is pushing down on your fingertip, and because you can respond, the muscle can contract, move out the way, and it will prevent any damage to your tissues. The next one I'd go for is transcriptional factors, and I'd use the example of estrogen binding to a transcriptional factor so that we could say that the receptor is the binding site for that estrogen molecule. And once that estrogen is diffused through the phospholipid bilayer into the cytoplasm, binds to the transcriptional factor receptor, it causes the DNA binding site to change shape. That entire molecule then moves into the nucleus, binds to the promoter region on DNA, enabling your RNA polymerase to attach, and then it initiates transcription. So that would be the receptor elements. And then when I talk about the importance of, I would link this to tumor suppressor genes. You can talk about this linked to cancer, because it's important that we are initiating the transcription of tumor suppressor genes, because those genes are responsible for making proteins that can reduce the rate of cell division to prevent uncontrolled cell division and therefore cancer. So that's an example that you could be talking about for that one. Or you could go for the other idea that it might be initiating the transcription of oncogenes, which means it's going to stimulate uncontrolled cell division, and link it to the concept of breast cancer. The next one I'm going to go for is chemoreceptors, and I talk about the fact that these are in the carotid arteries and they detect changes in carbon dioxide levels in the blood and therefore pH. And you could either talk about too high or too low a pH. I'm going to go for too low a pH, which would mean lots of carbon dioxide, which would be a result of exercise and therefore lots of aerobic respiration. So the chemoreceptors would detect that increase. That would then result in impulses being sent to the medulla, more impulses sent to the SAN along the sympathetic neurons, releasing acetylcholine, and that will then result in the SAN releasing that wave of depolarization more rapidly. The heart rate increases, and therefore blood is going to be delivered to the lungs and the alveoli for gas exchange, and therefore the removal of that excess carbon dioxide more rapidly. Now, the importance of that is it ensures that your blood doesn't become too acidic, and if that was the case, it could start to denature proteins, for example, hemoglobin in your red blood cells and enzymes, and that would affect the transport of oxygen and also chemical reactions. So that would be the concept for that one. The next one I'm going for is HIV, and for this one, you could talk about the structure of HIV. So you've got your capsid, and inside of that, you've got RNA reverse transcriptase. You've then got an envelope around that, which is taken from the host, and attachment proteins. Now, the receptor element here is the fact that those attachment proteins can bind to the CD4 plus receptors on T helper cells in the host. And then once those bind, it enables the HIV to inject or insert its RNA and reverse transcriptase into the host, which is the T helper cell. Now, I'm going to link this to the importance of this enables HIV to replicate, because once that RNA and reverse transcriptase have entered the host cell, the RNA is converted into a single strand of DNA through that reverse transcriptase enzyme. That DNA then enters the nucleus and is going to be incorporated into the host's DNA. So then the host will start to transcribe and translate viral proteins, which can then assimilate to make a new virus particle, which ultimately causes that T helper cell to burst. And the importance of here is it enables the HIV to replicate, and if enough T helper cells are destroyed, it's going to result in such a lowered immunity that the person will then develop AIDS. Now, importance doesn't always mean it is good. In this case, it's important because it's causing such harmful impacts. So importance doesn't necessarily mean it's a good thing. The last one I go for is osmoreceptors. So I talk about the fact that these are receptors for changes in the water potential of your blood, and they're found in the hypothalamus in the brain, and they can shrink and shrivel in response to the water potential. But let's go for the concept of your blood water potential is too negative. Therefore, water is going to leave the osmoreceptors by osmosis and they will shrivel. This will then result in more ADH being released or produced by the hypothalamus, sent to the pituitary gland, which then releases it into blood, transports in the blood until it reaches the receptors on the kidneys, which are the target cells. And then you can talk about how that results in more aquaporins being embedded into the walls of the collecting duct and the distal convoluted tubule. So more water is reabsorbed from the nephron back into the blood. And that is how you then get your blood water potential increasing back within that normal range. Now that is important because, and this is where you could then link it to why it's so important that we have enough water in our blood. Water is an important solvent so that it can transport substances around the body. It's also important in chemical reactions, so as a metabolite for hydrolysis and so on. So that's why it's so important that you have enough water in your blood. Now you could also have a whole range of other ideas here, because you could have had the beta cells in blood glucose as an example of a receptor. You could have also had, instead of the Pacinian corpuscle, you could have talked about the rods and the cone cells. Other examples of receptors where you've got binding together. So you could have talked about antibodies and antigens. So a whole range of things you could have said for this one. So there we have it. Those are my 3 plans for those 3 essays. I recommend you now have a go at writing those 3 essays in full. Because we've done the full plan already, I recommend you give yourself 35 to 40 minutes to write that essay. Remember, go for 4 or 5 of the paragraphs. And if you do want more help to make sure you get as high a grade as possible on this essay, because as we said, it's worth almost 10% of your A-level grade, then don't forget to check out my biology bootcamp in the description below, which has got everything you could possibly need to get you top grades. But for now, that's it, and I'll see you next week for more help with your A-levels.